Welcome back to Hidden Wisdom Academy Biology. Today, we'll explore the anatomy of Compositae and Gramineae, two plant families with unique internal structures. Compositae, or Asteraceae, is a dicot family with bicollateral vascular bundles. Gramineae, or Poaceae, includes monocots that show Krantz anatomy in their leaves. We'll see how their structures help in function, adaptation, and evolution. Also, we're close to our 100th episode. We posted a poll on our YouTube community. Vote and tell us what you'd like in the special episode. Now, without further ado, let's get started. Now let's look at the anatomy of the Compositae stem, which is a typical dicot stem. The outer layer is the epidermis, covered with a cuticle and bearing multicellular hairs. Just below it lies the cortex. The parenchyma gives strength, and the parenchyma helps in photosynthesis and storage. The endodermis is not clearly seen in most herbaceous stems. Inside, the stele has a sclerenchymatous pericycle and a ring of bicollateral, open vascular bundles. That means xylem is in the center and phloem is on both sides. Between the bundles are medullary rays for sideways transport, and in the middle, a large pith made of parenchyma completes the structure. Now let's study the anatomy of the Gramineae stem, which is a typical monocot stem seen in plants like maize or wheat. The outer layer, called the epidermis, has compact cells with a thick cuticle that prevents water loss. Inside, there's no clear cortex or endodermis. The whole inside is made of ground tissue. The vascular bundles are many and scattered throughout the ground tissue. Each bundle is surrounded by a sclerenchymatous bundle sheath for support. The bundles are collateral and closed, meaning they don't have cambium, so no secondary growth occurs. The xylem looks Y-shaped, and below the protoxylem, there's a small water cavity, a typical feature of monocot stems like maize. Let's now compare the stems of Compositae and Gramineae. In Compositae, like sunflower, the stem is dicot. It has a clear cortex, bicollateral vascular bundles arranged in a ring and show secondary growth because of cambium. In Gramineae, like maize, the stem is monocot. The vascular bundles are many and scattered, collateral and closed, and each has a sclerenchymatous bundle sheath. There's no secondary growth, and the inside is filled with ground tissue instead of separate cortex and pith. A water cavity below the protoxylem is a special feature of monocot stems. Remember, these key differences help you easily identify dicot and monocot stems under the microscope. Let's learn the Gramineae leaf anatomy, like in maize or wheat. The epidermis on both surfaces is covered with a cuticle. The upper surface has bulliform cells to help the leaf roll in dry conditions, and stomata are present on both surfaces. The mesophyll cells are tightly packed with few air spaces, chloroplasts are small without grana, and there's no distinction into palisade and spongy layers. Each vascular bundle is surrounded by bundle sheath cells, which are large, thick-walled, and rich in chloroplasts, the site of the Calvin cycle. Vascular bundles are collateral and closed. This special arrangement is called Krantz anatomy, meaning wreath in German. Let's compare dicot and monocot leaves. Dicot leaves, like sunflower, have mesophyll divided into palisade and spongy layers, no Krantz anatomy, and stomata mostly on the lower surface. Monocot leaves, like maize, have undifferentiated mesophyll, thick-walled bundle sheath cells around vascular bundles, bulliform cells on the upper epidermis, and stomata on both surfaces. This Krantz anatomy in monocots supports C4 photosynthesis, making them more efficient in high light and heat. Knowing these differences helps you identify leaves and their adaptations for exams. Let's recap the anatomy of Compositae and Gramineae. First, stems. Compositae has a cortex, ring-arranged bicollateral vascular bundles, and cambium, so secondary growth occurs. Gramineae has scattered closed bundles with sclerenchymatous sheaths, no cambium, and a water cavity. Next, leaves. Dicot leaves, like sunflower, have palisade and spongy mesophyll, stomata mostly on the lower surface, and no Krantz anatomy. Monocot leaves, like maize, have undifferentiated mesophyll, stomata on both surfaces, bulliform cells, and Krantz anatomy for C4 photosynthesis. Mnemonic, stems have RBC that is ring, bicollateral, cambium. Leaves have DIPA that is dicot palisade, one surface, MOKR that is monocot krantz, both surfaces. Remember these key points for exams. Knowing structure helps you identify and answer questions easily. Here's your challenge question for today. Assertion A. The stem of Compositae shows secondary growth, but the stem of Gramineae does not. Reason, our Compositae stem has bicollateral vascular bundles with cambium, whereas Gramineae stem has closed vascular bundles without cambium. Is it option A? Both A and R are true, and R is the correct explanation of A. Is it option B? Both A and R are true, but R is not the correct explanation of A. Is it option C? A is true, but R is false. Or is it option D? A is false, but R is true. Comment your answers or send them to us on Telegram or Instagram. That's a wrap on Compositae and Gramineae anatomy. Now you know the key differences between dicot and monocot stems and leaves, from vascular bundles to mesophyll and Krantz anatomy. In the next episode, we'll cover secondary growth in dicot roots and stems, how plants get thicker, the role of cambium, and the formation of xylem and phloem. Also, don't miss our daily NET MCQs and PYQs for quick revision. See you in the next episode.